Hi everyone, welcome back to this channel. In today's video we are diving into one of the most fascinating process in cell biology, that is mitosis. We will break down each phase step by step, help you to understand how cell divides and ensure the genetic continuity in next generation. Whether you are a student or just curious about life processes at cellular level, this video is for you. Mitosis is a process of nuclear division in eukaryotic cells in which equal distribution of identical sets of chromosomes occurs in two daughter nuclei. It is very important process and it has role in growth, development, repair of damaged cells into new cells, and because of this process organisms develop by asexual reproduction. It may be took place by mitosis or meiosis. Both are the types of cell divisions, and in today's video our main concern is on mitosis. So we will discuss about the changes that occurs in the cell or in the nucleus, majorly during karyokinesis. What are the changes that take place in the nucleus, or what are the points that occurs, the changes that are occurred in different phases of nuclear division, that is karyokinesis. Let's start with the first phase of nuclear division, that is prophase. In prophase, following four major changes occurs. And before diving into the phases or the steps that occurs in these phases, let me tell you one interesting fact about cell cycle. Before diving into a division phase, cell prepare itself for division, and that phase is very high metabolic rate phase, and that phase is called as interphase, but that phase is also misleadingly called as resting phase. Apparently no changes took place in that phase. Cell just make some protein, some duplicate its chromosome, and it is misleadingly called as resting phase, but the name of that phase is interphase. After interphase, mitotic phase start, and in mitotic phase division happened. First, in prophase, firstly cells chromosomes become starting visible. How it happened? Because of the condensation of chromosome, centrioles will move towards the opposite poles, and then nuclear membrane and nucleolus will disappear. Starting again, we will discuss about the four steps one by one. First of all, chromosome becomes visible with condensation, centrioles move towards the poles, nuclear membrane will disappear, and nucleolus also will disappear. These are the four changes that will occur in prophase of mitosis. After that, second phase will start, that is metaphase plate. In second phase, chromosomes align at the equatorial plate, forming a metaphase plate in a center. A plate will form in a center of the cell. That plate are called as metaphase plate. Spindle fibers will attach to the centromere with the help of kinetochore proteins. Because kinetochore proteins are also present in centromere and are also present on spindle fibers, because three kind of spindle fibers originate from centrioles, and that three out of three types, one centriole spindle fiber is called as kinetochore spindle fiber, and that will attach with kinetochore protein in chromosome. Alignment ensures equal division of chromosome in next generation, in next cell. Next phase is very simple, but it's very important and crucial, because this is the phase which ensures the equal distribution in next generation. Like sister chromatids are pulled apart towards the opposite poles, and equal distribution, or no disjunction, you can say it's a disjunction, equally disjunction in each cell's part is very important, and that's happened in anaphase. Centromeres split. Each chromatid is now considered as a separate chromosome, and it will move toward the opposite pole of the cell. And the next phase is telophase, which then ultimately leads towards the cell into a cytokinesis. In telophase, chromosomes decondense back into the chromatin, nuclear envelope reforms around the two sets of the chromosome, and spindle fibers disassembles. Telophase is just the reversal of prophase. All the changes that occurs in prophase, it will be reversed in telophase. After that, cell will divide. Division of cytoplasm will occur. In animal cells, cleavage furrow will form and the cell will pinches out. In the plant cell, it will be different. You can see in the animation how cell plate will form and cleavage furrow is happening from the center, but in plant cells, a cell plate will form, and that plate will helps in the formation of new cell membrane and cell wall, which then leading the separation of one cell to another cell. This is all about division. Can you imagine how much time is required by the first universal simplest cell of the life to complete this process? You can also answer me in comment box and I will check your answer. If you like the content, do like, share and subscribe and share it with your friends.